This video goes through the chapter 8 notes, which covers free energy and enzymes. So there's our focus, and we will cover enzyme structure, enzyme catalysis, and environmental impacts on enzyme function. This might be a long one. I'm sorry. Pause and take a break if you need to. Oh, and cellular energy. So, metabolism is the totality of an organism's chemical reactions. Each metabolic pathway shows the steps from starting molecule to product or products. Each step in the pathway is catalyzed or sped up by a specific enzyme, which you can see here. So from A to D, we have 3 different enzymes that do 3 different reactions. Catabolic pathways release energy by breaking down compounds. So an example of that is cellular respiration, where glucose is broken down. Anabolic pathways use energy to build complex molecules. And that an example there would be protein synthesis, when ribosomes are putting all the different amino acids together. Before we get into more stuff on that, let's talk a little bit about energy. Energy is the capacity to cause change, and there are different types. Kinetic energy is the energy of motion. Thermal energy is the kinetic energy due to random motion of atoms or molecules. And heat, which we talk about often, is thermal energy and transfer from one body of matter to another. Light energy can be harnessed to perform work, like during photosynthesis. And potential energy is the energy that an object possesses because of its location or structure. So you can think about maybe the water behind a dam has lots of potential energy. And finally, chemical energy is potential energy available for release in a chemical reaction. So this would be a catabolic pathway. Here's just a picture example of different types of energy. The diver on top of the platform has potential energy, and the diving person has kinetic energy. We also need to talk about the laws of energy transformation, which leads us into thermodynamics, which studies energy transformation. The first law of thermodynamics says that energy is transferred and transformed, but it can't be created or destroyed, which will be important later, as will the second law of thermodynamics, which says that energy transfer or transformation increases entropy of the universe. Entropy is a measure of our disorder or randomness. So spontaneous processes are ones that will occur on their own without any energy inputs because they increase entropy or disorder. Here's a picture example of the cutest little bear showing the 2 laws of thermodynamics. The first law, he says that energy is transferred or transformed. So energy from the food is going from the food source to the bear. And then the second law shows energy transfer or transformation, where heat is being released. Although order is a characteristic of life, we still want an overall increase of entropy in the universe. Gibbs free energy, or free energy, is the portion of a system's energy that can perform work when temperature and pressure are uniform throughout the system. And the change in free energy, or delta G, can be calculated. And its formula is delta G is equal to delta H minus T times delta S. Delta H is change in enthalpy, delta S is change in entropy, and T is the temperature in Kelvin. Any process with a negative delta G or negative change in free energy are spontaneous, meaning that they will just happen without anything else causing it to happen. So you can think of free energy as a measure of a system's instability, which means it will have a tendency to change to a more stable state. Systems will move towards greater stability, and as reactions move towards equilibrium, free energy of the mixture of reactants and products decreases. So free energy increases when the reaction is pushed away from equilibrium, and it will right itself until it is in equilibrium again. Because all reactions have a forward and reverse, and they will go whichever way needs to maintain equilibrium. So this diagram shows the relationship of free energy to stability, work capacity, and spontaneous change. Some examples there will be gravitational motion, diffusion, and chemical reactions. There are two types of reactions in metabolism, exergonic and endergonic. Exergonic reactions have a net release of free energy, which means they have a negative delta G value. Endergonic reactions absorb free energy, and so their delta G value is 0 or positive, which you can see in the diagram here. A is an example of an exergonic reaction. So it's starting, has lots of free energy, and then there's less at the end. So these reactions will release energy. Endergonic reactions, like in figure B, have to consume energy because the products have a higher free energy than the reactants. ATP powers cellular work by coupling exergonic reactions with endergonic reactions. Cells do three main types of work. They can do chemical work by pushing endergonic reactions when they synthesize polymers, like proteins. Cells also do transport work to pump substances across the membrane against the gradient, it's like active transport, and mechanical work, for example, beating cilia and muscle contractions. They are able to do this work by coupling exergonic reactions to drive the endergonic reactions. Since the exergonic ones have a negative delta G value, they're going to occur spontaneously and release energy. And so then that release of energy can power an endergonic reaction that has to consume energy to happen. So let's talk about our favorite energy molecule, ATP, for a second. ATP stands for adenosine triphosphate. It contains the sugar ribose, the nitrogen base adenine, and 3 phosphate groups. The bonds between the second and third, or the phosphate group that's on the end, are relatively easy to break, and they are broken by hydrolysis, which leaves us with ADP, adenosine diphosphate, plus a phosphate. The hydrolysis of ATP is an exergonic reaction that is often used to power an endergonic one. So here you can see the structure of ATP. We've got our 3 phosphate groups, a sugar, and an adenine. And then below that, you can see the reaction. We have the hydrolysis, and that's what we're left over with. Proteins in a cell can harness energy released during ATP hydrolysis to perform cellular work, which again would either be chemical, transport, or mechanical. Enzymes will use this energy to drive a reaction that is endergonic on its own. So the interactions are coupled so that it's overall an exergonic reaction with the coupling of the two. It will usually involve the phosphorylation, which is the transfer of an ATP phosphate group to something else, which could be a reactant, which forms a phosphorylated intermediate that is more reactive than the original molecule. So essentially what's happening there is one of those phosphates from ATP will break off and attach to something else, and whatever the phosphate has attached to has therefore been phosphorylized, and that phosphorylated intermediate is more reactive than the original molecule, so that will help that reaction to continue because we have something in a more unstable state. So here is a picture example of that. Transport and mechanical work are also powered by ATP hydrolysis. What will happen here is we'll have a change in the shape of the protein, which will alter its ability to bind something else. Recall that the structure of the protein will determine its function. So in the picture here for A, the with ATP attaches to that transport protein, which forces it to open and close to either side of the membrane. Or with mechanical work, an example would be binding to motor proteins that are hydrolyzed, which causes a shape change, and then it moves the motor protein on. Those are 2 examples of ATP hydrolysis for transport and mechanical work. The good news about ATP is that it is easy to regenerate. ATP is regenerated by the addition of a phosphate group to ADP. Free energy to phosphorylate ATP comes from other exergonic reactions in the cell. The shuttling of this phosphate and energy is called the ATP cycle, which you can see below. So we start with a little ATP and a little H2O. So that gives us energy for cellular work. Then we're left with ADP and P. And so then they will be phosphorylized, and then we're able to start the whole process again. Last, let's talk about a special class of proteins called enzymes. Enzymes speed up metabolic reactions by lowering energy barriers. So, although spontaneous reactions can occur without any outside input, they can be so slow that it takes forever for them to actually happen. And we don't want that to be the case. So, enzymes will speed up reactions that would have happened anyway. Enzymes are proteins that act as a catalyst. Catalysts are things that can speed up a chemical reaction without being consumed. And so that means that enzyme can do the same job over and over and over and over again and is not disrupted by whatever reaction it is catalyzing. Let's talk about the activation energy barrier. So, activation energy is the amount of energy reactants must absorb before a chemical reaction can start. A lot of times that is supplied by heat. When molecules have absorbed enough energy for bonds to break, the reactants are in a transition state, which is unstable. The formation of the new bonds releases more energy than was invested to break the old bonds, which is what we want. And most of the time, activation energy is so high that the reaction will not be able to proceed. So enzymes are able to lower how much energy it takes to get over the hill of the reaction, which you can see here. So we start with our reactants that have a higher amount of free energy than the products. They have to go up the hill to the transition state before they're able to make and break bonds, or break and make bonds, and then the products are formed. Catalysis is the process by which a catalyst selectively speeds up a reaction without being consumed. So enzymes catalyze reactions by lowering the activation energy barrier, as I said. And again, they just speed up reactions that would happen anyway, but a whole lot faster. Each enzyme is specific to the reaction that it catalyzes. Here is a graph showing the course of the reaction with and without the enzyme. The black line shows without an enzyme and the red line is with. So you can see that the overall delta G is unaffected, but the amount of energy it takes to go over the hill of the course of the reaction is much, much lower, which therefore will increase the speed. The substrate is the reactant or thing that the enzyme acts on. The substrate will bind to the enzyme to form the enzyme substrate complex. The region of binding where the substrate and enzyme actually touch is called the active site, and it will be a groove or pocket on the surface of the enzyme where catalysis occurs. While the enzyme substrate complexes form, the enzyme converts the substrate or substrates to the products or product. One thing we know about enzymes is that they usually end in the three letters ASE. For example, the enzyme lactase breaks down the sugar lactose, and that's all it does. Even though lactose is a pretty basic disaccharide, lactase does not work on other disaccharides. Enzymes can recognize its specific substrate even when there are similar compounds around, and that is because of the protein's unique 3D shape and how it is folded. The tightening or binding after the contact is the induced fit. So after we have that binding, we have a tightening of that interaction. So here is an example here. That big blue blob is the enzyme, and the little red piece is the substrate. It perfectly fits in on that active site, and then it will tighten to keep the substrate where it is. That's the induced fit. Substrates will be held at the active site by weak interactions like maybe hydrogen bonding or ionic bonding. The R group of the few amino acids at the active site are the ones that catalyze the conversion of substrate to product, and then the product leaves the active site. That leaves the enzyme free to work on another substrate, and this happens really fast. Some enzymes can work as fast as going through 1,000 substrates per second. Most metabolic reactions are reversible, again, because we need to be able to have that tendency towards equilibrium. So the enzyme might be able to catalyze the forward and the reverse of the reaction. And which way it's going depends on the relative concentration of reactants and products and what's needed. And so the net effect will always be in the direction of equilibrium, which you can see here. The cycle of the enzyme. We have bonding to form the enzyme substrate complex. We break the things and then release and then start all over again. The rate of the reaction depends on the concentration of the substrate. However, when the concentration of substrate is higher than how quickly the enzyme can work, then it's saturated. And so at that point, the only way to increase the reaction rate is by making more enzymes, which sometimes can happen. But we won't get to that until we talk about protein synthesis a bit later. How efficiently an enzyme can function is affected by environmental factors like temperature and pH. Each enzyme will have its own optimal condition where it will work the best. So, for example, our body temperatures are around 37 degrees C. So, most of the enzymes that live in the human body have an optimal pH of around 37. To a certain point, increasing temperature does increase the reaction rate, but then it will swiftly drop off because when it gets too high, the enzyme can denature or start to lose its 3D shape, and it will not be able to work as well. It's kind of a similar deal for pH. Most optimal values will be between 6 and 8, because that's generally where things are. There are some exceptions to that, like your stomach has a pH of closer to 2. And so those enzymes, like pepsin, that work in your stomach have an optimal pH of, closer to that. And they will not work when it is that basic, even though that's close to neutral. So here is an example of that. As you can see, pepsin, optimal pH around 2, and then an intestinal enzyme is going to be a little bit higher. A lot of enzymes will need non-protein helpers for catalytic activity, and these are called cofactors. They can be bound tightly to the enzyme or loosely bound and sometimes unattached. There'll be things like zinc, iron, and copper. Cofactors that are organic molecules are specifically called coenzymes. Some things inhibit enzymes from being able to do work, but that is not always a bad thing. If it is a competitive inhibitor, it blocks the substrate from entering the active site. So whatever the thing is that is inhibiting binds to the active site. Non-competitive inhibitors bind somewhere else on the enzyme that force it to change shape, which therefore changes its ability to function. You can see here. So picture A shows normal binding. B is an example of a competitive inhibitor where the active site is being blocked. Non-competitive inhibitor binds on the side of the enzyme opposite. That's not always the case, but in this example it is. And we have that shape change that prevents the substrate from being able to bind properly. We also have allosteric regulation of enzymes, and these molecules regulate enzyme activity. They change the shape and therefore function of the enzyme, but they do not bind at the active site. Allosteric regulation is the binding of some sort of regulatory molecule to a protein that affects its function somewhere else. So it's not binding at the active site, but it changes the active site's ability to do what it needs to do. In some cases, we have an allosteric activator that increases the enzyme's ability to do its job. Allosteric inhibitors prevent the enzyme from being able to do its job. Also need to look at feedback inhibition. where a metabolic pathway is stopped by the inhibitory binding of the end product of the enzyme. So this is an example of the feedback inhibition of isoleucine, which is one of the amino acids. So what happens is isoleucine, when there's lots and lots of it, can bind to an allosteric site on the enzyme to prevent it from from the pathway to continue so that we don't have more of it made. Just don't forget that cells are compartmentalized. Some enzymes and enzyme complexes will have fixed locations in the cell, and others are enclosed in specific organelles to give them the microenvironment they need to be able to do their job.